Aquí está.
Todos pueden, según yo.
Sí, creo que ya está grabando.
Sí, sí, ya me dice en la
Ok,
esquina que está grabando.
va. Pues bueno.
Okay. Okay. So the article I read is called DNA Methylation Signatures of Smoking and Lung Cancer is Enriched for Exposure Signatures in Newborn Adult Blood. So I'm going to do my best and explain a little bit on what this means and what these researchers did. So basically what they tried to do is to explain or to create a relation on DNA modifications, specifically methylations or hypomethylations, which is the removal of the methyl group in a specific site of DNA, and related to different types of not organisms, but like groups of people. In this case, there were smokers, like just normal people that smoked, smokers that had stage 1 cancer, non-smokers, uh, former smokers, and newborn babies which were exposed to to, to smoke in the uterus. So first of all, I'm going to explain a little bit on on some of the terminology that is important. So a CPG site is a region of DNA with where we have cytosine, and it's directly followed by guanine. Um, these can in the article, these are the things that are studied, and they're either methylated or hypomethylated, which is removed or added a methyl group. And this is important because this modification of a CPG site can affect the expression of a gene. So this is what these researchers studied. So using a tool called Illumina 450K beta ray, which is basically a tool that allows them to quantify and to observe these CPG sites. They observed these modifications throughout different groups of people. So one of the things they observed is that there was a great correlation between these groups that for example, they found a great correlation between people that had stage 1 cancer and were formerly there were former smokers, which indicates that there's an expression of genes, and there's a modification in how the body works and constantly expresses genes if there's like a symptom. So for example, these cancer patients with lung tumor presented a correlation of 0.75 out of 1 and of CPG sites with current smokers. And this like relation other um, between these groups were made, and they also found, for example, that one of the things that they found is that adult blood that smoke can be like has a very similar behavior to blood from a newborn that was exposed to smoke in the uterus, which is pretty surprising. It has a correlation of 0.49, and for example, they also explored how this methylation of these CGP sites affected other organs. So for example, they specifically focused on smoking, which is, well, obviously it affects the organ that is affected the most is the lungs, because there's direct exposure from the smoke and these methylation modifications that are made in DNA. However, these methylations affect the whole body overall, and one of the things that are affected the most or significantly is the blood. So, in general, when the blood is affected, the whole body is affected. And they studied how this modification of blood and how this change in the behavior of the blood affected other organs. And what they found is that smoking specifically and the methylations or hypomethylations that are affected or created by smoking and smoke exposure of cigarettes is mostly it mostly impacts the lungs. They observed how it impacts the bladder, and yeah, they found a very yeah, it did affect the growth of cancer and the proliferation of cancer and the malfunction of the body, but it didn't. The CPG sites didn't have a great correlations, which led them to believe that smoking specifically and the exposure to cigarette smoke doesn't affect the whole body, or it does affect the whole body, but it doesn't impact the rest of the body as much as it does to the lungs. So it is pretty amazing to find these things out. They also found out, or not found out. They could also um, assume that some of the symptoms caused by cancer and and some of the symptoms caused by smoking in general are attenuated by time. So, for example, former smokers had very similar modifications to former smokers had very similar CPG site modifications to the ones in in newborn babies. So these babies had never smoked; they just had been exposed to all of the chemicals in cigarettes and to a bunch of things. And former smokers showed a very similar modification, showing that yes, symptoms are are or symptoms from smoke exposure are attenuated, but uh, and yeah, some symptoms are attenuated, and they can. It's not like they're cured, but they can be left behind, and they're not permanent. So yeah, that's pretty good. And uh, in general, yeah, and that was pretty much it. It generally talks about how some how the exposure to smoke and all these modifications does affect um, the tissue, for example, the lungs, as I was saying, and some affects the organism overall. But yeah, yeah. Do you have any question? I know it's like a little bit complicated, and I didn't understand it all, but overall, it's pretty cool how we can relate to all these things.
I have one question.
Hmm.
So, this paper is about how smoking relates to lung issues or to whole body issues, right?
Yeah,
Uh,
it
is there any like chemical that's shared not only by cigarettes but by anything else that you smoke? That's like a common factor between everything. And that's
well, what do you mean?
like something that many things that people smoke, like weed or something like that, um, may have in common with cigarettes, with tobacco.
Yeah, yeah. Well, it doesn't specifically say what chemicals. It mainly focuses on tobacco and how tobacco and mainly like normal cigarettes, not like cigars, normal cigarettes with a bunch of different chemicals that are just in there, are exposed to, or how we are exposed to those chemicals. Even they even included some people that weren't smokers at all. They just had like normal smoke exposure from the city, from friends, and all that stuff. And they did show some modifications in the DNA, that were similar to the ones of former smokers. So
Okay.
yeah.
So thank you. That was my question.
Another question? Yeah, no?
No one?
I
think
don't know
Victor.
if you said it, but what were the difference in the DNA exactly?
Oh, the difference in the DNA. So there's these groups that are called cytosine. They're abbreviated by the CPG sites. So it's a cytosine followed by a guanine, and these sites are methylated or demethylated. So there's either an aggregation or a removal, and this modification it changes the gene expression of something. So this gene expression can vary from uncontrolled growth or the loss of something, or the loss of a function and things like that, which can lead to cancer. And it is observed in most of the subjects, because they were mostly, or it was targeted for smokers. Like the study was targeted at smokers. So yeah.
Okay.
So any other question?
Mm-mm.
Okay. So I guess we can move on to the next one.
Okay, well, I'm going to share a presentation. Mhm. Did you see it? Yes.
Yes.
Yeah.
Well, the article's name is Correction of a Pathogenic Gene
Yes,
Mutation
I see
in
it.
Embryos. What? Okay. Yes. And basically, it is about the correction of the heterozygous MYBPC3 gene that has a mutation and corrected in the embryos when they are in the single cell stage. And basically, they used CRISPR-Cas and homology direct repair. And well, this gene this gene could express like monogenic autosomal dominant mutation, which cause problems in the heart, like in the thickness of the ventricles and basically problems in the heart, and that can cause death, basically. But it is very hard to know if you had the mutation, because the symptoms are presented like not in the early stages of your life, like when you're older. So basically the article talked about the way they make an experiment to prevent or to correct this mutation, and like the steps they had to take in order to have the best outcome. and to try it in vivo, because this was an experiment that was tried in vitro. So the individuals they made in this experiment were heterozygous, and the sperm was a carrier, while the oocyte, that is the female gamete, it was the wild type and didn't have the mutation. The mutation has a deletion that is shown here, the GAGT deletion of the MYBPC3 gene. And the experiment was to target the deletion in the single cell embryos. So they use CRISPR-Cas signaling RNAs and templates of a DNA that wasn't the DNA of the body, which has like 2 different base pairs to differentiate it from the DNA of the body. That one of these base pairs code for an enzyme that is a recognition site of these like different template. The transcription efficiency was of 27.9%. So they proved like 2 ways of CRISPR-Cas, the CRISPR-Cas1 and the CRISPR-Cas9 2. And they decided that they were going to use CRISPR-Cas9 because it has a higher homology direct recognition efficiency. So basically, yes, the experiment was made by an injection of the Cas9 protein, the signaling RNA, and the templates of DNA after 18 hours of fertilization. And it showed that 66.7% were homozygous wild type, and 33.3% were mosaics. This was a problem because mosaics cannot live, cannot be transmitted and cannot be developed into embryos. So here's a picture of the process when the CRISPR-Cas was injected and only lead to wild type embryos. But this was with a control sperm that has that doesn't have the mutation. And in the experiment, they did again the CRISPR-Cas9, but with a mutated sperm and non-mutated oocyte. In the S phase, is important to remember that this experiment was done in the S phase. And there were 3 outcomes on target mutant embryos that didn't change with the CRISPR-Cas9. They stayed wild type mutant. Also, they had this mosaic embryo that has a wild type mutant, like the target embryos, the repaired embryos, and other type of cells, I don't know, other type of repaired, with non-homologous enjoining. And the other outcome that was the homology direct repaired embryo. So the researchers believe that the problem was in the phase that they inject the CRISPR-Cas, because when they made the injection in S phase, because of the process that is occurring, they had 2 mutants, and CRISPR-Cas remain active and will continue targeting these mutants, and made a cell with with different outcomes that lead to mosaicism. So they decided to inject the CRISPR-Cas, like the Cas9 protein, the signaling RNAs, and the SSODN DNA templates with the sperm, but in the M phase, in the mitotic phase of the oocyte, in order to have only one outcome. And they discovered that it had a 100% efficiency, while in the S phase it had a 72.2% efficiency. Efficiency, I mean in no mosaicism embryos. So here are the results of the S phase embryos, and you could see like there are a lot of mosaics and non-homologous enjoining, which cause partially the mosaics. And when they changed the experiment with in the M phase, they had basically all the outcomes were uniform embryos that were repaired or wild type mutants. And that was experiment, and they discovered that there wasn't normal development of the embryos, no abnormalities, and no indels detected. Like the indels are the mosaics and the non-homologous enjoining, like there are mutations, and they found that there were no mutations after the procedure. And basically that's all, because they discovered that all the cells, or basically all the cells turn into blastocysts, and well, a big percentage of them, and that the process was like, well, right, and has no problem, but they still do not they still decide to not try it with real embryos or in vivo, because there are a lot of things to continue researching, like in all the topics. But they thought that this was a very good like approach to prevent or to correct the embryos before they born, and to target all these mutations that are dominant. And basically that's it. Do you have any questions?
I don't know if you explained it, but which is the reason for why it works 100% efficiently in the M phase embryo than the S phase?
Because, well, I did not explain it, but I think they did not like explain it in the article, but I think that in the S phase is where the replication of the DNA is occurring. So you have two DNAs in the cell, and you are like changing two genes in one cell. So in the mitotic phase, you like they are getting separated, and you can have only like you modificate only one DNA in one cell and one on the other. I think that's the reason. Like you don't have the
Okay.
two genes in one cell.
Okay. Yeah, it makes sense.
Yes. Another question?
So where was this experiment done?
When?
Where?
Where? I don't know. They don't say anything. I think United States. I don't know.
Víctor, se ve que quieres preguntar algo.
Sigo pensando en.
Well, there are a lot of Chinese names in the persons that write the article.
I heard people were more permissive in China regarding CRISPR-Cas9
Maybe.
last time.
I've also read that they're allowed to use CRISPR-Cas on a lot more organisms than in anywhere else in the world. So it's pretty crazy how that changes between regions and how like we can do some more researches in other places.
So
Was
maybe
this?
that's.
No.
It's very likely for it to be like a Chinese team or something like that.
Yeah, maybe.
But Victor was saying something.
No.
What do you think about this article, ethically speaking? Like,
Well,
correct.
I think that there's like no real ethical problems because it was no in vivo. But if they perform it like in a real human, I don't know, because like it's to correct it to. I don't think that it has to be like not optional. So if the parent wants to correct its baby, I don't think that's unethical. But well, yeah, I don't know.
I mean, correct your baby.
What?
I mean, if you had a baby, would you apply something like this? I think that's a real question. No?
Well, yeah, but I don't know. I think that yes, I would correct it. Or well, it depends on the person because some people aren't like prepared to have a baby with a deficiency, and like it's also okay to decide what do you want.
I think that's correct. I mean, if you're already having a baby, then why not assure he or she has a good life?
Mm-hmm. Yes, because they had like a ton of like heart diseases when they grow up, and I think that's a worst life that try to stop it and try to make it better.
I agree.
Yeah, I agree. But okay, so this is a little bit different. So do you think something like this for like CRISPR-Cas to repair or to change and correct like an error should be implemented into like a public healthcare system thing, or it should be like a private thing?
Like
That
a personal interest.
yeah,
I
like
think,
if you can afford it, you can do it.
well, I
Well,
think
I
that
think it's like.
uh-huh. Well,
Well,
I think that
I
nowadays
was.
we don't because uh, of its novelty, like it's really new and really expensive nowadays. should be like a private thing, but when the science advance and all the things like are more controlled and you can have like cheaper this process like in a cheaper way, I think should be implemented in a public place because it's really expensive. Like if you implement it right now, it wouldn't be like gaining anything, and the science of this process could not advance as fast if you pay for it. That, in contrast.
I think it may be like, as Victor was stating the question, as a public health matter, it would be like a good strategy for assuring the health of the future populations. Still, it may be very expensive, if I'm not wrong,
Yeah.
and it probably would be only implemented in super rich countries with super small populations.
Yes.
Yeah, I think so too. I mean, there are some basic treatments right now which are they're expensive, but they're not like astronomically expensive, and we can't give that service to everyone. So I don't think we'll be able to give this embryo correction for
Well, imagine how like people died from practically everything,
oh yeah.
and maybe in another 100 years, I don't know.
But
Yeah.
also, I think science grows like exponentially. So like before, what was not affordable was thought to be affordable in 100 years, like we think of it now, but it only took them 10 years to be affordable. And now that same technology has advanced and it now takes 5 years to be affordable, and so on. So maybe technologies like CRISPR-Cas that are now taking, well, many scientists are taking advantage of it in many ways, like we've seen in 2 articles already. It is probable that it is possible that it becomes available to the public faster than we may think. I don't know.
Yes, but
Yeah.
to make it public, like you have to wait until it's affordable.
Yeah,
So
that's true.
I think it will be affordable, maybe in, to say a number, 20 years.
Yeah, but besides it being affordable, I really don't. I really think there's a lot of people that will say that it's very wrong to do it. And many,
Oh yeah, definitely.
like many countries would be com- it, and maybe only countries like China, for example, would allow
Mhm.
because they're more open-minded on these types of
Yeah, I
research.
think it's a very delicate debate.
yeah.
And I think that it's have to good use because not only for the look that you have, you can change your baby. I think this method is good for um, health or things like that.
Yeah, I mean, the use is very important. I can see some people just using it instead of like health, just because
Mm.
I want to be, I want my baby to be this tall, or I want my baby to be this color, or blah blah blah.
Oh yeah, but that's different. Yeah.
Yeah, yeah, yeah. But it's an issue that could come up
Yeah.
or will come up with this technology.
Hmm.
Mm.
But yeah,
So yes, we have like less than 1 minute.
Okay.
Mm.
Okay.
Good.
mm.